My name is Ron Mitsui. I'm director of the UCLA Center for Clinical AIDS Research and Education. My background is that I'm a hematologist, oncologist, and an immunologist, and I've been involved in both AIDS patient care and clinical research since 1982. My initial introduction to the AIDS epidemic was in seeing patients with Kaposi sarcoma. And then over the years, my interest in the immune manifestations of AIDS kind of got me more involved. And then once the disease was found to be virally related, we started looking at the interactions between viruses and immune system, and that's how I really got involved in this epidemic.
What were the results of such trials, and have all or some of these drugs, therapies, been legalized since the trial?
Well, here at the UCLA Care Center, we've been involved in clinical trials of a number of antivirals. The majority of antiviral studies that we've been involved in have been with the so-called nucleoside analogs. This is a group of drugs such as AZT, DDI, and DDC. These studies were sponsored by the National Institutes of Health through a program called the AIDS Clinical Trials Group, of which we're one of 44 centers around the country conducting trials in both adult and pediatric HIV patients. The initial studies were with AZT in both the AIDS patients, the ARC patients, and then the asymptomatic. And these studies demonstrated that AZT not only slowed the progression of the disease, but increased CD4 numbers and ultimately resulted in longer survival for patients with AIDS. The next drug we studied was DDI, and these were done in comparison to AZT in patients who have failed AZT therapy. And then the third drug that had been in clinical testing was a DDC, and these studies have been done both as monotherapy studies, if you will, one drug, versus combination therapies with AZT or DDI. And we're now involved in several studies comparing these various drugs and or combinations of these drugs to see which is maybe more effective and in various populations. Patients that have not had prior treatments, patients that have had a lot of prior treatment, and then patients with differing CD4 numbers. So it becomes quite complex, as you can imagine, and involves a number of centers because no one center can accrue all the patients you need to answer definitively questions of efficacy, that is, whether the drug is active and has effectiveness in delaying the progression of AIDS. The clinical studies so far have demonstrated that AZT is clearly effective, DDI is effective, and is probably most effective in patients that have failed or progressed on AZT. DDC has been shown in the past to be effective in combination with AZT, but not as effective as a single drug in patients that have never been treated with an antiviral. Just recently, last week, the results of another clinical trial that was done in a national group suggested that DDI may be as equivalent to DDC in its activity in patients that have had prior AZT therapy and who seem to be not responding. So the field is changing all the time, and what we're faced with as physicians as well as researchers is deciding how best to treat any one patient. And this becomes a very complicated area and is the reason why we're doing clinical trials.
Could you explain the process in which trials are conducted?
Well, clinical trials are a development of the science of medicine over the last few years. And the way drugs are licensed in this country anyway is that it first has to be shown to be safe, and we determine that in what we call phase 1 testing. Then we go to a second study that shows activity, that is, it must be shown to do what we think it should do. So in the case of an anti-HIV drug, we would have to be able to show that it suppresses HIV and that it increases T cell numbers. Then the third step is the phase 3 studies where we actually compare the drug that we're interested in against standard therapy. And this may involve several hundred to several thousand patients and may take a year to two to complete. But in this case, what we're hoping to demonstrate is true efficacy. That is, it does in fact improve the well-being of the patient. And that's why it takes so long because of the statistical natures of these studies to prove that one drug may be more effective than another.
What are the phase 1, 2, and 3 trials?
Well, just in a quick sense, phase 1 studies are safety studies. We treat a small group of patients with differing doses of the drug until we get to a maximum tolerated dose. And this is defined as that dose in which we see toxicities that would limit us from going to a higher dose. Okay. Once we've determined that, then we can move on to what we call phase 2 trials or activity studies. In these studies, we look at a fixed dose typically, 1 or no more than 2 doses, and treat a group of patients to determine what proportion of patients will truly benefit from the drug. Now, in some cases, these phase 2 studies are randomized study with a placebo control. In other cases, they're just straight-out treatment studies with no placebo. And a phase 3 trial is the next step in which we compare the drug that we're interested in against standard therapy to see whether the drug is more or less effective than what we would typically treat a patient with. And if there is improvements, then that would then become the new standard of care. So it's an evolutionary process that is not only important in drug licensing, but also in terms of helping us determine how best to treat patients.
What does these different phases mean for getting access to a particular drug or therapy?
Well, the FDA requires that a drug be found to be safe and effective before they'll license it for general use. In order to do that, a drug would have to get through phase 1 and through phase 2 testing. It doesn't necessarily have to go through phase 3 testing, but in general, phase 3 testing are initiated before the drug is licensed, so that at some point we'll have some data to compare the drug in question with standard treatment. Okay? It typically takes, for most drugs, about 5 to 6 years from the time the phase 1 studies are initiated to the time a drug is licensed. And overall, it may take 10 to 12 years from the time a drug is discovered till the time that it's licensed. The FDA has made a very concerted effort to try to speed up this process, specifically for drugs that are useful in AIDS and cancer and other life-threatening diseases. But it still is a very long and involved process, and the reason is that we need to be sure that a drug is not only safe but effective, and that we're not giving drugs to patients that have no activity or they may cause harm.
What are the latest trials you're conducting or know of?
Well, we're doing a number of studies with new antiviral drugs. These include drugs that are not only belong to the same class of drugs, the so-called nucleoside analogs, as AZT, DDI, and DDC, but also drugs that belong to completely different classes of agents. And these other drugs may act in a completely different way from the currently available drugs. So there's a possibility that these new drugs may be more effective and may also prevent the development of what we call resistance to the currently available drugs. So we're involved in, for example, studies of drugs like D4T, which is a new nucleoside analog, as well as other drugs which belong to completely different classes, such as the TAT antagonists, which is drugs that inhibit the TAT gene product, protease inhibitors, which inhibit the protease enzyme. We're looking at vaccine therapies, which act by stimulating the immunity against the virus and virally infected cells. And we're also involved in a study with a drug called CD4 PE40, which is a conjugate of CD4 with a toxin which is targeted at HIV-infected cells and designed to kill it. So there are many different groups of drugs that we're looking at. Most of them are in what we call phase 1 testing, but some of the more exotic drugs, if you will, we hope to get through phase 1 testing within the year and into the phase 2 studies.
What have been the results?
Well, as I indicated, most of the drugs are early in their development, so it's way too early to say whether they're going to be active and effective. What we can say is that many of these drugs are very well tolerated. They all have their side effects, but in general, at the doses where we can see some activity against the virus, they tend to be reasonably well tolerated, and in many cases better tolerated than drugs like AZT. So that's very encouraging, and what we hope is that as we bring more of these drugs into larger studies, we'll be able to find more effective treatments that will be less toxic and that we can use either alone or in combination with these other drugs.
Have there been any changes in getting drugs or therapies approved by the FDA which might change the need for such trials?
Well, I don't think we'll ever get away from doing clinical trials because it's the best way, in my opinion, it's the best way that we have to determine that a drug is in fact safe and effective in people. You can do lots of studies in animals, you can do lots of test tube studies, but these will just demonstrate that the drug has some activity. In order to prove that it really is effective, you have to treat patients. And the only way that you can prove that a drug is effective in a large number of people is to test it against either the best standard approach, best standard drug available, or against a placebo. So while the process for conducting these studies and approving drugs has been speeded up, I don't think we'll ever get away from doing clinical trials. And in fact, as a physician as well as a researcher, I would want to make sure that I knew that a drug had activity and was safe in a group of patients before I actually treated my patients with it.
What problems are there with these trials?
Well, obviously, with any kind of new drug, you're putting patients at risk for side effects. Some of them we know, or we suspect we know what they might be from animal studies, but in other cases, the side effects that we see may be completely unexpected. For example, the problem with pancreatitis with DDI was completely unexpected until they got to clinical testing in man. Similarly, one cannot predict whether a drug will induce peripheral neuropathy based on animal studies or laboratory studies. So whenever you test these drugs in people, you're putting patients obviously at risk for these side effects, some of which may be very serious and life-threatening. Clearly, we don't want to do that unnecessarily. So before we will treat patients, we want to have as much data as we possibly can from animal and laboratory studies, and we want to proceed carefully through these various stages of testing in order to protect patients from unnecessary risks. We also will monitor patients very carefully. That's the major problem with clinical trials testing. There's a lot of unknowns. Also, when you get done with a study, you have to be certain that the data you have is valid. So a lot of energy, a lot of effort is put into the design of a study even before you treat the first patient, because you don't want to get through a study involving, let's say, a couple hundred patients and find that for design reasons the trial was completely invalid. Then you've wasted patients' time, their efforts, put them at risk unnecessarily. So there's a science actually involved to developing these studies, and it's something that we do here at UCLA and at other research centers, and why the National Institutes of Health, for example, has set up this trials group. The purpose being to design trials that will answer questions definitively so as not to put patients at unnecessary risk, and also to make sure the studies are done efficiently.
What is the future for AIDS treatment?
Well, if you ask me to look into my crystal ball, what I see coming down the pike is a large number of drugs, some of which will prove to be useful, as others of which will not be useful, and that at least in the next 5 years or so, we'll find drugs that are more effective, perhaps less toxic than currently available therapy, and we'll be able to prolong the lives of AIDS patients several-fold. Now, if you're asking me, will there be a cure for AIDS, I would say probably not in the next 5 years or so. There's a lot of hope being put at the moment into something called gene therapy, These are approaches that actually inhibit the production of virus at a genetic level. And this may be the only way that we can effectively control the virus from making more virus once it's already infected the host. For patients that are not infected, the best hope lies with vaccines, which may prevent people from actually acquiring the virus in the first place. So I think there's a lot of hope and a lot of promise based on what we're now learning about the virus and from a lot of the scientific work that's being done in the laboratory. But I don't see an immediate cure, at least in the near future. But that's not to say that I'm pessimistic, because I think that we have learned a lot, and there are some very promising areas of research that I think will lead to very effective control of the virus and hopefully a cure, if you will, or at least a way of inhibiting the virus from doing destructive things to people that are already infected.
Finally, is it going to be easier to gain more access to drugs therapies in the future?
Well, I would hope so. The limiting feature at the moment is the cost of doing these trials. It's very expensive to conduct clinical trials. We obviously do not ask patients to pay for participating in research studies, since we're learning about the effectiveness of these drugs in the process of doing these trials. On the other hand, it becomes a very expensive proposition to treat hundreds of patients and to do all of the monitoring that's necessary to prove that a drug is effective. Now, in the past, pharmaceutical companies have paid for a lot of that. Also, the federal government, through these National Institutes of Health grants, have provided the bulk of the funding for research. And as we all know, research funding is slowly drying up because of the federal deficit. We also know that pharmaceutical companies are under a lot of pressure to reduce their costs, and I think rightly so. And that means that there's going to be a squeeze on the research dollars available to us. Obviously, I think as a researcher, we would love to have unlimited amounts of money in order to treat patients and conduct these trials in a very expeditious way. But I think that where we're going to be faced with limitations will be in the availability of resources to do these studies.
Do you want to talk about ozone and do you know about passive immune therapy?
Yeah, I know. Oh, sure.
Can you tell us what you know about alternative treatments a little bit? What you think of ozone, hyperbaric oxygen?
All right. Well, in talking about alternative therapies, you have to define your terms. I think alternative therapies, to me anyway, are those therapies that are being tested in patients without a lot of either laboratory or animal data to support their effectiveness prior to initiating those trials. Many of the drugs that are being conducted or being tested in what we call the underground, if you will, are just such drugs. That's not to say that they're ineffective. It just means that there's less data to support their effectiveness before you launch into these clinical trials. Now, there are some dangers involved in that there may be more unexpected side effects. There may be adverse effects of the drug, such as stimulation of virus as opposed to inhibition of virus. There may be adverse effects involved in the interaction of the drug you're testing with other drugs the patient may be on. And so there are lots of variables that may not necessarily be incorporated into the design of your trial and accounted for, which could ultimately lead to some problems for patients. That's my major concern with alternative therapies. Now, you mentioned a few of these so-called alternative therapies, such as hyperbaric oxygen, ozone therapy, and so on. These therapies would fall into that category, since they're being used based on some theories that haven't been fully tested, at least not fully tested in laboratory and animal models prior to going into patients. Now, in the past, a lot has been learned from those kinds of serendipitous observations that doctors or scientists have made, and then very bold physicians that have gone forward and tested these things in people. In most cases where a drug is in fact going to be found to be useful, those early observations should then lead to more rigorous scientific evaluation of the drug, which will prove definitively that that drug is useful. And those of us that are involved in the scientific testing of these drugs in clinical trials are very open-minded. We're always willing to listen to people, to test drugs for them in the laboratory, and if in fact if the data is convincing, then there's not one scientist worth his salt that I would think would say that we shouldn't test those drugs in patients. But I think it needs to be done in a very careful way so that we don't put patients at unnecessary risk. Specifically with the ozone and hyperbaric oxygen, I know of the theory behind them, but I have heard very little in terms of the clinical trials results. Hopefully those will be forthcoming.
What was the other thing you mentioned?
Passive immune therapy.
Yeah, passive immunotherapy actually has quite a bit of scientific basis for its utility, and may in fact be the basis for the role of vaccines in treatment of patients who are already infected with HIV. What we mean by passive immunotherapy is taking plasma from patients that are HIV infected, which contain high titers of antibodies against the virus involved in this epidemic.
What were the results of such trials, and have all or some of these drugs, therapies, been legalized since the trial?
Well, here at the UCLA Care Center, we've been involved in clinical trials of a number of antivirals. The majority of antiviral studies that we've been involved in have been with the so-called nucleoside analogs. This is a group of drugs such as AZT, DDI, and DDC. These studies were sponsored by the National Institutes of Health through a program called the AIDS Clinical Trials Group, of which we're one of 44 centers around the country conducting trials in both adult and pediatric HIV patients. The initial studies were with AZT in both the AIDS patients, the ARC, but not as effective as a single drug in patients that have never been treated with an antiviral. Just recently, last week, the results of another clinical trial that was done in a national group suggested that DDI may be as equivalent to DDC in its activity in patients that have had prior AZT therapy and who seem to be not responding. So the field is changing all the time, and what we're faced with as physicians as well as researchers is deciding how best to treat any one patient. And this becomes a very complicated area and is the reason why we're doing clinical trials.
Could you explain the process in which trials are conducted?
Well, clinical trials populations are patients that have not had prior treatments, patients that have had a lot of prior treatment, and then patients with differing CD4 numbers. So it becomes quite complex, as you can imagine, and involves a number of centers because no one center can accrue all the patients you need to answer definitively questions of efficacy, that is, whether the drug is active and has effectiveness in delaying the progression of AIDS. The clinical studies so far have demonstrated that AZT is clearly effective, DDI is effective, and is probably most effective in patients that have failed or progressed on AZT. DDC has been shown in the past to be effective in combination with AZT, but yes, my name is Ron Mitsui. I'm director of the UCLA Center for Clinical AIDS Research and Education. My background is that I'm a hematologist, oncologist, and an immunologist, and I've been involved in both AIDS patient care and clinical research since 1982. My initial introduction to the AIDS epidemic was in seeing patients with Kaposi sarcoma. And then over the years, my interest in the immune manifestations of AIDS kind of got me more involved. And then once the disease was found to be virally related, we started looking at the interactions between viruses and immune system, and that's how I really got in patients and then the asymptomatic. And these studies demonstrated that AZT not only slowed the progression of the disease, but increased CD4 numbers and ultimately resulted in longer survival for patients with AIDS. The next drug we studied was DDI, and these were done in comparison to AZT in patients who have failed AZT therapy. And then the third drug that had been in clinical testing was a DDC, and these studies have been done both as monotherapy studies, if you will, one drug, versus combination therapies with AZT or DDI. And we're now involved in several studies comparing these various drugs and or combinations of these drugs to see which is maybe more effective and in various